hybridization, actually still the same topic. Okay, so we have something called hybridization. Okay, if you do hybridization, you can like visualize the protein or DNA or RNA. Okay, so they are more detailed than gel electrophorescence, of course. Gel electrophorescence is just using agarose gel, separating them. But if you want to do it more detailed, higher resolution, separate them better, you have more choice here. We can even have other like gene chips, so have a fast detection of a lot of different type of gene at the same time. Okay, so some practical available technique that you can consider. Okay, so we can study the protein, we can study the DNA, and so you have all these tools. So what we can do about with this, we can do some DNA fingerprinting by checking the blood, but of course better by checking the DNA. So if we want to do DNA fingerprinting, what are the available techniques we have? We have to actually in the lab, you try to do the RFLP on the traditional Chinese medicine. So that is one way. So you should know about the principle. You use precision enzyme, you cut them up so that they become different fragments. But of course, first you have to extract the DNA, purify the DNA, amplify the DNA, and then you start with precision enzyme. You have to make sure you have enough DNA to work with first. So if you after PCR you have enough DNA, you do the precision enzyme digestion to cut them up, and because if there's different species, they have different sequence, so they will be cut in different point into different fragment. So the products, the resulting products of cutting by precision enzyme will be different. You want a gel, they will have different bending pattern, and this is called precision fragment length polymorphisms, what we call a RFLP. Okay, then you want a gel, you will be able to tell that they are different. Okay, so some key steps shown here, you have done it, you have do similar thing already. You have the SDS paste gel, you have precision enzyme digestion. Okay, so basically it's like this. RFLP, the whole step of our LP. So make sure you know the key steps, what we are doing with this analysis and why we can tell the result by this. And we also have something really more use on human DNA fingerprinting. Is making use of the variable number tandem repeats. And the most commonly used one is the short tandem repeats. There are some short fragment repeat a lot, but repeat in different numbers in different individuals. So we can tell that they are different individuals by checking how many repeat number they have. Okay, so if you are doing this STR analysis, what we call, the key step, the preparation step is the same. Obtain DNA sample, purify them, amplify them, and then apply something, and then check the bending pattern at the end. Okay, so still, gel electrophorescence, visualize the band, compare with reference or compare with each other. So no matter which type you are using, the principle are the same. Okay, but you have to know RFLP is because you cut them into different segments, and then for STR is that they have different number of repeats, so they have different size, different length of the DNA fragment, so you can tell the difference. Okay, so these two are commonly used DNA fingerprinting techniques, and with these tools we can make use of them for forensic, finding the killer, the murderer in a crime scene, or for paternity test, finding the true mother or true father, and of course we can have DNA fingerprinting, maybe not these two techniques but more techniques on other applications like species identification on non-human, on like checking the food, whether they are the meats they claim to be, they are beef meat or they actually are chicken, which is cheaper. We tried this already. We can check the authenticity of the traditional Chinese medicine. Okay, because some are really expensive, but some are much cheaper. So we have to check whether they are the real one, the authentic one, or the fake TCM. We can also check for check the eDNA from environment to find the hidden species. You cannot really see them. You cannot really collect the species, but they are there. Then if you can find the DNA, you can confirm they are there in the environment. So this is more on the conservation part, but this is really a application that we are using today with the technique we've had. Okay, and then a lot of bioethics, so I'm not going into detail again, but for every application you can mention, you should also be able to think of at least one possible bioethics. Okay, are there any drawback? Are these techniques affecting anyone, or will they damage the natural environment, something like that? So be able to think about the related bioethics. Okay, you have the techniques, you have the application, you have the advantage, but there are always some drawback or some concern you have to think about. Maybe you can solve it, but still you have to think about a conscious decision that you know about the risk and concern when you're applying a biotechnology here. Okay, and that is your another concern about you. You are the concern here. Okay, and lastly, Kongling. So a very huge debate on bioethics is the Kongling on human. So just one example for you to think about. And then so we have covered all the 9 lectures. So just a very short recap on the lab session also. Okay.